Good day, this is Professor Will, CBMD PhD, and today is Monday, July 19th, 2021. It is 1:00 and 48 seconds a.m. Eastern Standard Time. Today's research, cancer cells. So let's hop to it here. Quantum physics helps destroy cancer cells. Researchers have found a way to enhance radiation therapy using novel iodine nanoparticles. Date July 14, 2021. Source, Kyoto University. Cancer cell death is triggered within 3 days when X-rays are shown onto tumor tissue containing iodine-carrying nanoparticles. The iodine releases electrons that break the tumor's DNA, leading to cell death. Exposing a metal to light leads to the release of electrons, a phenomenon called the photoelectric effect. An explanation of this phenomenon by Albert Einstein in 1905 heralded the birth of quantum physics. And the research provides evidence that suggests it is possible to produce the effect inside cancer cells. A long-standing problem with cancer radiation therapy is that it is not effective at the center of tumors, where oxygen levels are low due to the lack of blood vessels penetrating deeply into the tissue. X-ray irradiation needs oxygen to generate DNA-damaging reactive oxygen when the rays hit molecules inside the cell. Researchers have been trying to overcome this issue by finding more direct ways to damage cancer DNA. In earlier work, researchers showed that gadolinium-loaded nanoparticles could kill cancer cells when irradiated with 50.25 kiloelectron volts of synchrotron-generated X-rays. In the current study, researchers designed porous iodine-carrying organosilicon nanoparticles. Iodine is cheaper than gadolinium and releases electrons at lower energy levels. The researchers deciphered their nanoparticles through tumor spheroids, 3D tissue containing multiple cancer cells. Irradiating the spheroids for 30 minutes with 33.2 kilovolts of X-rays led to their complete destruction within 3 days. By systematically changing energy levels, they were able to demonstrate that the optimum effect of tumor destruction occurs with 33.2 kilovolt X-ray. Further analysis showed that the nanoparticles were taken up by the tumor cells, localizing just outside their nuclei, and shining just the right amount of X-ray energy onto the tissue-prompted iodine to release electrons, which then cause double-strand breaks in the nuclear DNA, triggering cell death. The study represents an important example of employing a quantum physics phenomenon inside a cancer cell. It appears that a cloud of low-energy electrons is generated close to DNA, causing double-strand breaks that are difficult to repair, eventually leading to programmed cell death. The team next wants to understand how electrons are released from iodine atoms when they are exposed to X-rays. They are also working on placing iodine on DNA, rather than near it, to increase efficiency and to test the nanoparticles on mouse models of cancer. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.